Hello and welcome to this lesson on the action potential, a very important and basic process in neuropsychology. The action potential is the mechanism by which cells in the brain and nervous system communicate with each other. It's a process that involves chemistry and biology, so we will be highlighting some information from both of those disciplines in this video. Also, please understand that this is a very simplified process that I'm going to explain. And what actually happens in the brain is a lot more complicated because it involves multiple cells acting in concert and on a single neuron. So we're really going to get very simple here, but hopefully it'll give you the foundation that you need to be able to learn more about neuroanatomy as you go through the course and as you go through your learning about neuroscience in general. So here we go. This is a neuron. A neuron has several parts that you need to be aware of. First are dendrites. Dendrites are these protruding, kind of purplish finger-like projections that come out of the cell body. They receive the signal from other neurons. This long piece that's coming out of the neuron that has these kind of red things on it is called the axon. The axon, most neurons just have one axon, although many in the spinal cord have multiple axons, but for our purposes we're only going to focus on ones that have one axon. These conduct the information from the cell body to the axon terminals, which connect to the synapse, which we're going to speak about here in just a minute. All along the axon is the myelin sheath, which is a fatty layer, as it says there, that speeds transmission. We'll talk about the nodes of Ranvier and Schwann cells here in a bit. So here is a very simple neural chain. So here you have the previous axon connecting to the dendrites of this middle neuron here, and then this middle neuron then connects to the dendrites of the next neuron. And that's basically how most of these processes go. One neuron communicates with another neuron, which communicates with the third neuron. So in the synapse, which is the space between neurons, no neurons touch, by the way. All neurons have this tiny space in between them, and that is where the main chemical reaction starts. So you see these little black dots. These are called neurotransmitters. Neurotransmitters are chemicals. And as you know with chemicals, they bond to things and create reactions. And so that's basically what's happening here. So one neuron is going to release its neurotransmitters into the synapse, and they're going to bind with the receptors on the receiving cell. Okay, so in order for anything to happen in a neuron, you have to have a certain threshold that is met. So let's say that you need 100 neurotransmitters to create a reaction. That is your threshold. So let's say you only get 50. You are not going to have a reaction. Okay. Let's say you get 150. That'd be great, right? You'd maybe get a stronger reaction. No, actually, you wouldn't get any stronger reaction than if you got 100, if you got 150. You just have to meet this threshold in order for there to be a reaction. This is called the all or none principle. Okay. That's really important. So what's happening here is that in the cell, you've got an overall negative charge. In fact, the charge inside the cell is negative 70 millivolts. Okay, and so all inside here is negative, and mostly outside the neuron, you have a generally positive overall charge. Okay, so what happens when these neurotransmitters bind with this cell? These receptor sites here make the cell wall semi-permeable. That means that some of the ions that are inside, remember they're overall negative, and remember it's overall positive out here, they want to get out, okay? Because remember, in chemistry you have this principle that things that are in areas of greater concentration want to move to areas of lesser concentration. So there's a lot of pressure inside this receiving neuron and they want to get out, but they can't, because for most of the time the cell wall is not permeable, meaning things cannot move freely in and out of the cell wall. So once these chemicals kind of rest on there, it kind of creates this reaction where the receptor sites open up and allow some of these negative ions to move out of the cell, and some of these positive ions move into the cell. So within these chambers, you've got ions, charged particles, moving in and out of the cell. So this creates an electrical reaction, which is the action potential. So within each receptor site, you've got this exchange of ions, and it creates this little electrical charge that kind of causes then the next chamber to open up and more ions move freely back and forth. And it moves all the way down the cell wall, all the way down there. So you see this electrical charge kind of gradually moves down the cell wall because these ions are rubbing up against each other. And the rubbing up against each other creates electricity, and the electricity then kind of continues the chain reaction all the way down the cell. Once it hits, though, the axon, you've got this myelin sheath, which is a really cool thing. Because as you kind of notice, this is a pretty slow process, because every single site along the wall has to be activated, and the ions have to rub together in order for the electrical charge to be maintained. Well, that's not very efficient. So what happens when you get to the axon is that it's covered in this nice fatty substance that causes this electrical reaction to not have to go through every channel in order to be maintained. So it goes really quickly all the way through here, through the axon, all the way through. Now, remember I told you I'd talk to you about the nodes of Ranvier and the Schwann cells. So on this neuron, imagine that each chamber, each little gateway on the cell wall is happening. It gets all the way here to the axon, and it goes really, really fast right here. Well, these nodes of Ranvier are the spaces in between the myelin cells, or the Schwann cells, which make the myelin. And so the charge then gets to be maintained right here, right there in the node of Ranvier, and it makes, I'm sure, a noise just like that all the way through until it gets to the axon, all the way to the end. So the myelin sheath, in essence, acts like rubber tubing around an electrical wire. If you had an electrical wire that was not covered by rubber tubing, then the charge would just be kind of flying off of that electrical wire, and it wouldn't be very efficiently connected. It's kind of like if you ever have an appliance or a TV or something and the rubber tubing on the electrical wire is frayed, you don't get a consistent electrical signal being sent to your device. So you lose some of the charge if it's not covered by that insulator. So myelin sheath is your body's natural insulator. It helps to protect that axon so that the charge is maintained, and the charge actually goes much more efficiently through the axon than it did through the cell body. Now, as a side note, people who have disorders like multiple sclerosis have a condition in which their body is attacking the myelin, which is why people have trouble with movement and different kind of behaviors in that regard, because the myelin is not there to help conduct the signal efficiently. So myelin, therefore, is very important. So here you have the signal going all the way down the axon, and it finally gets to these terminal buttons here. And what it does once it gets here is that the charge acts as a push for all of these little synaptic vesicles that are in the synapse that contain the neurotransmitters. And they push these synaptic vesicles all the way down to the cell wall. And then once they get here, they get spewed out into the synapse, where then the process then starts all over. So, to kind of review, you have dendrites in the cell body, and here they receive the signal. You have this process by which an action potential is created, and it moves all the way down the cell until it gets to the axon. It shoots the information very quickly and efficiently down through the axon to the axon terminals, where the neurotransmitters are then spewed out into the synapse, and you have then conditions for a signal to be carried on to the next neuron. Now, what happens when all that's over? Because really, you have to have a time in which your neuron resets to that overall negative 70 charge, remember? So what happens is, as soon as the electrical charge moves through the membrane and makes the next chamber open to create the ionic charge, the first one is going to close up. And as they go along, they're going to close up in a chain reaction, just like they did when they were opening. And something called the sodium-potassium pump is going to pump the negative ions back into the cell and the positive ions out of the cell, so that you can get back to that overall negative 70 charge and create conditions in which it can now create an action potential. This is called the refractory period, the time in which the neuron actually cannot create a charge. It has to rest. It has to regroup, get its overall negative charge settled, so that it can be ready to create the electricity with the ions moving back and forth. So this is important because that way your system doesn't get too overloaded by too many action potentials happening at once. Now, just as a note here, this is the process by which excitatory neurons communicate with each other. Excitatory neurons are excitatory neurotransmitters are ones that create action potentials. There are inhibitory neurotransmitters that prevent a neuron from firing. So if you're wanting to calm down, instead of being revved up in response to stress, you might have inhibitory neurotransmitters that prevent your cells from firing, so that your behaviors then don't occur. So this process is for excitatory neurotransmitters and not inhibitory neurotransmitters. But it's still important to know that if a neuron is going to fire an action potential, then it has to have that threshold of excitatory neurotransmitters in the synapse in order for it to work. So this, ladies and gentlemen, is a very simple presentation of how a neuron works. Feel free to rewatch it as you need, so that you can have the information that you need for the AP exam. Good luck, and see you next time.